Okay, let's unpack this.
Let's do it.
We are diving into a really exciting intersection today. How advanced computational power, molecular AI and machine learning is fundamentally and rapidly transforming our ability to deal with global health threats.
Yeah, specifically thinking about pandemic preparedness and structure-based drug discovery. It's moving so fast.
It really is. So for you, our listener, we're peeling back the layers on two remarkable kind of connected stories.
That's right. The first is really about the product. It's about building a significantly better therapeutic countermeasure. We're talking a highly potent, highly resilient combination of monoclonal antibodies, MABs, effective against that whole family of coronaviruses called sarbecoviruses.
Right, the ones like SARS-1 and SARS-CoV-2. And the second story is, well, it's the engine that helped build that therapeutic and maybe what comes next.
Exactly. It's the introduction of this cutting-edge AI foundation model named Pearl.
Pearl.
Yeah, and it's currently setting, like, the new comp standard for predicting these precise 3D structures.
Which are absolutely essential for designing any drug based on structure, right?
Absolutely. That's the cornerstone. So our goal today is to show you not only what the latest defenses look like, but also how the underlying tools scientists are using are evolving at, well, breakneck speed.
Making the whole discovery process faster, more accurate, and way more adaptive than anything we had before.
Precisely.
Okay, so let's start with the immediate challenge. These sarbecoviruses, SARS-CoV-1, SARS-CoV-2. They pose this constant threat, partly because they jump from animals, but also, critically, our current antibody treatments, they struggle. The virus just evolves so quickly.
Yeah, variants pop up that just, get around the treatment.
Yeah.
We saw exactly that happen in real time with the Omicron variants. That's immune escape right there in action.
So scientists knew they had to find a way to sort of stabilize and boost the power of existing antibodies that target the bits of the virus that don't change much?
These broadly neutralizing antibodies, or BNAbs, they target those highly conserved areas, the parts that hopefully don't mutate as frequently.
So they picked two starting candidates. They showed some promise, but frankly, they needed a massive performance boost.
Right. The first one, CV325, it targeted a really critical conserved spot, the S2 stem helix. That's deep in the machinery the virus uses to fuse with our cells.
Okay, so a good target.
But its neutralizing power was, well, suboptimal. It wasn't strong enough.
Got it. And the second one?
That was S2X259. It targets a different spot, the RBD class 4 region. Now, this one was highly potent against the early strains of SARS-CoV-2.
But then Omicron came along.
Exactly. It completely lost all activity against later Omicron lineages like BA.45. The key just stopped fitting the lock, essentially.
Okay, so this is where human intuition kind of steps aside and the machine learning takes over.
Pretty much. They use this sophisticated structure-conditioned ML design approach. You can kind of think of it as guided evolution on a computer.
Guided evolution, I like that.
Yeah, it involved multiple rounds of design. The AI suggests millions of possible antibody sequences, and then these sequences are trained and filtered using other predictive models they call CQ-Func.
CQ-Func, sequence to function.
Exactly. Imagine protein design is like sculpting a really complex key. The CQ-Func's model quickly learns the rules of the lock the virus and the key material of the antibody.
So it helps the AI iterate faster, not just randomly, but towards sequences that are likely to actually work.
Precisely. Ones that will probably fold correctly and function powerfully. And this whole process led to two really remarkable new candidates, GB0669 and PRO37587.
Okay, tell us about them. GB0669 first.
Right, so GB0669 was optimized from that anti-S2 antibody, CV325. And it showed an immediate, really impressive 5-fold jump in binding affinity, how tightly it sticks to the SARS-CoV-2 spike protein compared to its parent.
Wow, 5 times tighter.
Yeah, and it also proved highly effective against the wider sarbecovirus family, including the original SARS-CoV-1.
So broader and stronger. How did the ML do that structurally?
Well, the analysis suggests the optimization basically stabilized the antibody's binding loops, the bits called CDRs, making them favor the bound shape. It sort of presets the key to fit the lock better.
Okay, that makes sense. Now, what about the other one, PRO37587? You said its story was maybe even more impressive.
I think so, yeah. Because its parent antibody, S2X259, was totally useless against variants like BA.45 and XBB.1.5, dead in the water.
Right.
But the optimized version, PRO37587, successfully rescued that neutralization activity. It worked against all of them.
Okay, that seems like a huge leap if the original was completely inactive.
This is where the structural insights get really fascinating. They use cryo-EM, this high-res imaging technique, and revealed how PRO 37587 pulled it off. The ML-designed antibody can actually accommodate a significant structural shift in the virus's receptor binding domain.
A shift caused by mutation.
Exactly. Specifically near a notorious Omicron escape mutation called S371L. That mutation essentially changed the shape of the keyhole on the virus.
Ah, so the old key, S2X259, couldn't fit anymore.
Right. It metaphorically snapped off trying to turn it.
Right.
But the new key, PRO 37587, designed by the AI, was sculpted differently. It could handle that structural shift.
Oh, does it just bind somewhere else?
Not exactly. It seems to harness these more complex binding mechanics, maybe things like allostery, where binding at one spot influences another spot, effects that are almost impossible for humans to just rationally engineer. The AI found a solution we likely wouldn't have.
That's really the power of the ML then, finding these non-obvious solutions.
Exactly. But the ultimate punchline here comes from synergy, combining the two new antibodies.
GB0669 and PRO 37587 together.
Yes, they work powerfully together because they target two completely separate sites on the virus. GB0669 hits the S2 stem helix, stopping the virus from fusing with the cell. PRO 37587 hits the RBD class 4 region, stopping it from grabbing onto the ACE2 receptor in the first place.
So distinct mechanisms, a dual attack strategy.
Precisely. And this dual attack led to enhanced potency across current Omicron variants, like XBB.1.16.1 and BA.2.86. But the really critical result, the defining one, was resilience.
Okay.
In a lab experiment designed to force the virus to escape an in vitro escape experiment using live Omicron BA.1 virus.
Yeah.
The combination showed no complete or partial escape through 10 full passages.
Wait, zero escape. Through 10 rounds of trying to force it.
Zero escape.
That is genuinely impressive. But, how reliable are those lab dish escape experiments compared to what happens out in the real world with millions of people and constant evolution?
That's a really crucial question. And you're right, in vitro experiments aren't a perfect crystal ball for nature.
Right.
But showing zero escape over 10 passages is a very strong indicator.
Yeah.
It suggests a fundamentally high barrier to resistance. Previous single antibodies often failed this kind of test after just a few passages.
Okay, so it's a significant hurdle for the virus.
A very significant hurdle. And importantly, this resilience translated into better protection in living models, too. The combo significantly reduced viral load in hamsters and special mice infected with different Omicron variants, much better than either antibody alone. It shows the ML design wasn't just good in a dish, it's robust.
Okay, so that antibody combination is an incredible achievement. That's the product of ML. But if that's what last year's tools could deliver, what's happening in the computational lab right now that could top it? Which I guess brings us to the next generation of these discovery tools, Pearl.
Exactly. We shift from the really successful therapeutic to the potentially groundbreaking technology that could enable future breakthroughs even faster. We need better, faster, more accurate prediction models.
So what exactly is Pearl? And, people have heard of AlphaFold cracking protein folding. Why do we need a new model? Is it just tweaking things or something genuinely different?
I'd argue it's a paradigm shift, actually. Pearl, which stands for placing every atom in the right location.
Catchy name.
It is. It's a generative model, but it focuses specifically on protein-ligand cofolding.
Cofolding,
Meaning, while AlphaFold predicts the shape of a single protein on its own, Pearl jointly predicts the three-dimensional structure of that protein when it's complexed with a small drug molecule. It predicts the exact binding pose, how the drug sits in the protein pocket.
Oh, okay. And that specific interaction is what matters for drug design.
It's everything for structure-based drug design, SBDD. Knowing that precise pose is critical.
Now, you often hear the big problem in molecular AI is just not having enough training data, right? We don't have billions of solved protein drug structures like image models have photos.
That's the core challenge.
Yeah.
And Pearl tackles this head-on. It augments its training not just with the limited experimental data we do have from places like the Protein Data Bank, but with a massive scale of synthetic data.
Synthetic data, so computer-generated examples.
Essentially, yes. They fed Pearl billions of calculated virtual examples. These are derived from existing structures, but introduce huge chemical diversity. It's like filling in the gaps where experimental science hasn't reached yet, exposing the model to a much wider chemical universe.
That sounds powerful. What about the AI's architecture? Is there some secret sauce there that makes its predictions better?
There is a key innovation, yes. Pearl uses something called an SO3 equivariant diffusion module.
Okay, that sounds technical. What does that mean in practice?
Right, forget the jargon for a sec. What it means is the model inherently respects 3D rotational symmetries. It's like teaching the AI the basic physics of 3D space from the get-go.
So it understands how molecules behave when they tumble and rotate.
Exactly. Because it understands that core physical law, the structures it generates, aren't just statistically likely based on the training data, they're also much more likely to be physically and chemically plausible. That drastically improves how well it generalizes to new unseen molecules.
Okay, more realistic predictions. And what about for the drug designer in the lab? You mentioned Pearl supports controllable inference. How does that help them get results faster?
Yeah, that's really practical. It uses this multi-chain templating system. So say you're trying to design a new drug. You can actually give the model some extra information to guide its prediction.
Like what?
Like the structure of an older related drug molecule, or maybe a known part of the binding pocket you want to target. You're essentially telling the AI, start your search in this specific area, or build something similar to this.
So you can incorporate existing knowledge directly into the prediction.
Exactly. That makes it immediately useful in real-world drug discovery campaigns, not just a theoretical tool.
That is practical. But does all this extra power mean it takes, like, a supercomputer weeks to run one prediction?
Yeah.
Is there a big computational cost or time trade-off for this accuracy?
That's always a concern. The paper suggests they focused on efficiency alongside accuracy. Incorporating those physical principles, like the SO3 symmetry, actually seems to make it more efficient in some ways because it generalizes better, reducing the need for just brute force searching.
Okay. And the proof is in the pudding, as they say. How does it actually perform?
Well, the results are pretty groundbreaking across key benchmarks.
We need atomic accuracy for this kind of work, right?
Yeah.
Measured in angstroms.
Precisely. That's the currency here.
Yeah.
So on generating accurate binding poses, meaning the prediction is within 2 angstroms, or 2A, of the actual experimental structure, which is a key industry threshold, Pearl delivered a 14.5% improvement over the next best models on a standard public benchmark called runs and poses.
Okay, 14.5% better at hitting that 2AA target. And I saw something about physical plausibility, too.
Yes, that's important. Its success rate barely drops when you apply strict chemical validity checks. It means the poses it generates generally make chemical sense. They aren't just statistically close, but physically impossible.
Right. They look like real interactions.
Exactly. But where it really shines, I think, is when you look at the high accuracy level needed for medicinal chemistry decisions. We're talking predictions below 1 angstrom, 1 istrope. That's the fine-grained detail you need to optimize drug molecules' affinity.
Okay, the super precise level.
Yeah. And on challenging real-world drug targets, using an internal data set they called internal XTEL's Pearl, achieved a nearly 4-fold relative improvement over comparable baseline models at reaching that sub-1x accuracy.
Whoa, okay. 4 times the success rate at that really tough, high-precision threshold.
4 times. That's the kind of leap that can genuinely transform how medicinal chemistry is guided.
Right, so now we can really tie the 2 threads together, can't we?
Yeah, I think so. In our first segment, we saw this incredible output, that dual-targeting antibody combination against sarbecoviruses.
The product.
That was the result, the maximum potency and resilience they could achieve using, let's say, the previous generation of ML-guided design tools.
And then in the second segment, we looked at Pearl. That's the cutting-edge tool, the future foundation, maybe. Achieving atomic-level accuracy nearly 4 times better on these tough targets.
And this convergence, this improvement in the tools, is fundamentally changing the speed of discovery.
How so?
Well, being able to model these complex protein-drug interactions with such high fidelity means future therapeutic candidates can potentially be designed much faster and maybe with higher certainty right from the start. You can anticipate things like those conformational changes, those binding effects, before you even synthesize the molecule in the lab.
So it shifts drug discovery further away from just trial and error in the wet lab?
Exactly. More towards simulation and prediction first. Because if these models can accurately simulate the precise structural ways a virus might try to escape, like that keyhole shift near the S371L mutation we talked about, then scientists can try to program resilience against those escape routes into the next generation of antibodies right from day one. You potentially skip years of iterative lab work, optimizing against variants after they emerge.
You design for resilience up front.
That's the goal. The antibody story shows us what early ML could achieve to tackle a pandemic crisis. The Pearl story, I think, shows us the roadmap for a future where designing, say, a pan-pathogen therapeutic might become a campaign measured in months, not years.
Because the AI is exploring billions of possibilities with much greater chemical and physical certainty.
Right.
And while that leads us right to our final thought for you, the listener, to maybe explore on your own, the highly resilient dual targeting antibody combination we discussed today, that was optimized using earlier generations of machine learning.
Mm-hmm.
So if models like Pearl, which are hitting almost 4 times the high accuracy success rate of previous comparable models on really challenging drug targets, if that is now becoming the foundation of molecular discovery, what new pathogens are we potentially ready to neutralize much faster? How drastically will that timeline shrink between seeing a novel threat emerge and deploying a highly effective atomic-level therapeutic countermeasure designed to anticipate its escape?
It's a powerful thought, isn't it? Really speaks to the state of scientific readiness that's developing.
It really does. Thank you for diving deep with us today.
My pleasure.